Theme: Epithelial tissues, simple epithelia. Plan: Number 1. General characteristic of epithelial tissues. Number 2. Morphologic and ontophylogenetic classification of epithelia. Number 3. Structure, localization, functions of simple epithelia. Epithelial tissues consist of cells arranged in continuous sheets in either single or multiple layers. Epithelial tissue forms covering and linings throughout the body. Epithelium is never covered by another tissue, so it always has a free surface. Epithelial tissue is present in two forms. The first one, as sheets of contiguous cells that cover the body on its external surface and line the body on its internal surface. And number 2, glands, which originate from invaginated epithelial cells. Epithelia are derived from all three embryonic germ layers, although most of the epithelia are derived from ectoderm and endoderm. Epithelial tissues have functions: selective barriers that limit or aid the transfer of substances into and out of the body, absorption of material from a lumen, for example intestinal tract or certain kidney tubules, secretory surfaces that release products produced by the cells onto their free surface, protective surfaces that resist the abrasive influences of the environment, and detection of sensations via taste buds, retina of the eye, and specialized hair cells in the ear. General morphological signs of epithelial tissues. Because the cells are closely packed, there is no extracellular matrix among them. Epithelium is located onto sharply marked basement membrane. Epithelial tissue is avascular, blood vessels are absent. Epithelium is characterized by sharply marked polarity of cells or layers. And epithelium has high regenerative capacity. Nutrition of epithelia. Because of epithelium is avascular, the blood vessels that bring in nutrients and remove wastes are located in the adjacent connective tissue. Exchange of substances occurs by diffusion through the basement membrane. Basement membrane consists of amorphous substance and fibrillar structures. Carbohydrate, protein, lipid complexes in basement membrane provide its selective permeability. Ontophylogenetic classification of epithelia. This classification is based on source of development. According to source of development, there are 5 types of tissues: epidermal, derived from ectoderm; enterodermal, derived from endoderm; celonephrodermal, which source is mesoderm; ependymoglial, derived from nerve tube; and angiodermal, which source is mesenchyme. The ectoderm gives rise to the oral and nasal mucosa, the cornea, the epidermis of skin, and the glands of the skin and the mammary glands. The endoderm is source of the liver, pancreas, and lining of the respiratory and gastrointestinal tract. The mesoderm is source of the uriniferous tubules of the kidney, the lining of the male and female reproductive systems, the endothelial lining of the circular system, and the mesothelium of the body cavities. Morphological classification of epithelia. This classification is based on shape of cells and relation of them to basement membrane. According to this classification, there are 2 types of epithelia: simple and stratified. In simple epithelia, a single layer of cells are present and all the cells lie on the basement membrane. In stratified epithelia, few layers of cells are present, and just lower layer of cells lies on the basement membrane. Simple epithelia are divided into squamous, columnar, cuboidal, and pseudostratified. Stratified epithelia can be keratinized, non-keratinized, and transitional. Simple epithelia also can be unilined and multilined. In the unilined epithelia, cells have equal shape and their nuclei lie on the same level, forming one line. In multilined epithelia, cells have different shape and height, and so their nuclei lie on different levels, forming few lines. So this epithelium seems like stratified. Another name is pseudostratified epithelium. Now about different types of simple epithelia. Simple squamous epithelium, mesothelium. Source of development is mesoderm. Localization, serous layers. Structure consists of mesotheliocytes with twisted edges containing 1 or 2-3 nuclei. Function, excretion and absorption of serous fluid, providing of mobility of internal organs, prevention from development of commissures. In this slide you see mesothelium impregnated by silver with staining of nuclei by hematoxylin. Numerated by 1, twisted edges of mesothelial cells are visible. Simple squamous epithelium, endothelium. Source of development is mesenchyme. Localization, blood and lymphatic vessels. Chambers of heart. Structure consists of squamous endotheliocytes, poor in organelles and containing pinocytotic vesicles. Function, exchange of substances and gases between blood and surrounding tissue. Simple cuboidal epithelium. Source of development is mesoderm. Localization, renal tubules. In this slide you can see numerated by 4 lumens of renal tubules and numerated by 3 their nuclei. Structure consists of cuboidal epitheliocytes with brush border in apical part and basal striations in basal part. Function, reabsorption of substances from primary urine into blood. Simple columnar epithelium. Source of development is endoderm. Localization, organs of gastrointestinal tract. Mucous layer of stomach, small and large intestine, gallbladder, ducts of liver and pancreas. Structure. In stomach, tissue consists of columnar epithelial cells producing mucous secretion, protecting lining stomach. In small intestine, columnar epitheliocytes contain brush border which participate in parietal digestion and absorption of products of splitting of food. In this slide you can see jejunum. Arrow shows microvilli forming brush border. Pseudostratified ciliated epithelium. Source of development is precaudal lamina of ectoderm. Localization, air passages, nasal cavity, throat, trachea, bronchi. Structure, ciliated, goblet, basal, intercalated, endocrine cells. Function, cleaning of inspired air from foreign particles, secretion and movement of mucus by ciliary action. In this slide you can see epithelium of trachea. Cells are visible, numerated by 3 ciliated cells and numerated by 2 goblet cells, producing mucinogen. Thank you for attention.